The following featured presentation is part of the Skywalking Network.
Welcome to Star Warsologies, the podcast about science and other academic fields of study seen in Star Wars. My name is Melissa Miller, and I am the science and technology writer at Nerdist and also a freelance contributor to Star Wars Insider magazine.
And I'm your co-host, James Floyd. I also freelance for Star Wars Insider magazine and StarWars.com. This episode, Star Warsologies, is going to chat about molecular and cellular biology as represented in Star Wars with cloning and genetic manipulation, and of course, everyone's favorite midichlorians. Our guest expert today is Dr. Karim Si-Tayeb, who is a former molecular and cellular biologist who now runs a book and game store in Nantes, France.
That's correct.
That's correct.
Melissa. Hi, James.
Hi, welcome.
Karim.
So it's really good to have you here, and I'm excited to hear more about that arc of your career later on in the episode. But for now, can you tell us a little bit more about yourself and quickly define maybe for us what molecular biology is and how that's different from cellular biology?
So I'm a former researcher in molecular and cell biology, working on human stem cells. And actually, the cell biology part is really focused on the cellular as an organisms that we can grow in culture and manipulate and try to challenge them to study diseases or developmental cues. And the molecular part is really inside the cells, trying to manipulate either the code with the DNA or the proteins that result from this code in order to change the messages that are inside the cells and between all the cells.
Okay, so that sounds like molecular is just stuff inside the cell and cellular is the cell itself.
Yeah, and then there is this communication between cells that control their behavior and how they get organized all together.
What's your history with Star Wars? Is it something you saw as a kid? Did it influence your decision to study this field of biology and genetics at all?
Yeah, so it's definitely something that I saw as a kid, and it was like an amazing experience because of this projection out of space on a huge screen. It's like pushing the limits of dream and exploration. At the time, so the scientific part was mostly on the spaceships, lightsaber, and droids. And it's later than the biology came along with episode 1.
Right, that's true.
Okay. Well, speaking of episode 1, one of the things that they bring up in that film is midichlorians being microscopic life forms that live in our cells and help us connect to the Force. Are there anything like that in real life?
So, yes, it looks like midichlorians were like an analogy of what is already existing in our living cells, which are mitochondria for the animal kingdom and chloroplast for the vegetal kingdom. And there are kind of symbiotes that are inside our cells and will allow us to manage with energy. And it's very different from mitochondria and chloroplast. The midichlorians have the same role with this interface that's going between life forms and the cosmic force. There was a lot of debates because in the field of Star Wars, because it looks like introducing this scientific explanation of how the Force will behave was maybe a negative way to explain it. But after, there was a lot of authors that used it actually to enhance the universe and try to explain also things that were not clear, like how Anakin is, why is he here, and how we can use that to manipulate the Force.
I definitely had that feeling. I know when I watch episode 1 now, is like, did we need this explanation? But at the same time, knowing more about how the Force works is really interesting. And what you talk about with plants and chloroplast, I'd never really thought about that before, but they say the Force flows through everything, right? So it would have to be in plants as well.
Maybe it's not. I don't know if there's anything on it in Star Wars, but how the definition of it is that all living forms possess those midichlorians. It should be inside all the vegetables as well.
I've
Wow.
never thought
So
of that before.
I know. What if you have a plant that is really high in midichlorians? It could have a very high force ability, but if it's not sentient at all, it could just
That's
be a
the thing.
super force powered plant. And I don't know, maybe if you ate it, it's like a force vegetable that can boost your abilities. Or maybe not. Maybe it would just be like, oh no, we're carrot midichlorians and we'll just be digested normally. But yeah, it's an interesting thought that plants
maybe
and the force.
There is like a real biology behind this, and as in those two different kingdoms that are animals and vegetables, maybe midichlorians does not behave the same way in animals than in vegetables.
Yeah.
Or even in larger like bacteria or other thing, fungi and stuff as well.
So now I am definitely picturing like bacteria with little tiny lightsabers.
Do bacteria have mitochondria?
No, because it looks like the hypothesis so far is that probably the bacterial world enter into the eukaryote world, this symbiosis, and the mitochondria or chloroplasts are actually close from bacteria, but they are not bacteria, because they have their own genetic information inside the chloroplast or the mitochondria. But some of those information along the evolution was lost, and the DNA from the host cells is filling the gaps to maintain the mitochondria or chloroplast. Okay. So then, just looking at the size, it's not possible for bacteria to have mitochondria or chloroplast. For more evolved organisms have mitochondria or chloroplast, yes.
So I know, going off of midichlorians, I never actually put together how similar those words are before somehow. But certainly George Lucas, read half of an article about midichlorians.
Probably, yeah.
Well, midichlorian is kind of a combination of mitochondria and chloroplast, that you just take the beginning of each word. It's midichlorian. Yeah. Whoa, we just discovered, most people get the mitochondria aspect. I think we just discovered the chloroplast aspect.
Yeah, I'm
you
sure
heard it here, folks.
So going off of that, what's your theory of that whole Anakin's paternity, which I'm not sure I needed an explanation. And I'm in episode 1 when he was like, immaculate conception sort of thing. I'm not sure I needed that. I'm curious if there's any science behind this now.
Well, maybe, but it's really hypothetical. But in the episode 1, Darth Sidious claimed that Anakin may have been the result of Darth Plagueis, his master, experience. And that maybe his mother has been inoculated with something that gave life to a baby that have huge amount of midichlorians. So an aspect that was interesting in those episodes is how they detected those the midichlorians levels, because it was with a simple blood sample that they may evaluate the count of midichlorians. But what is not clear, because there is this definition that midichlorians may be a symbiote that are inside the cells, but other things can make us think that it's something that is also flowing in the blood as a microorganism. Or if we consider that it's inside the cells, they may produce proteins that are then detected in the blood, and that will allow them to estimate the level of midichlorians. So it's not clear.
Okay.
Well, I mean, we know that the Palpatine family isn't like against abandoning kids on desert planets, right? That happens to Rey too. So, this idea that Shmi is, some sort of experiment and then just tossed aside or something like that is, I don't know, that actually seems pretty reasonable to me.
Well, is she really tossed aside, or is it a way of like, let's have the experiment, but keep it out of Republic space, and if something happens, we can go pick up the kid, and if nothing happened, then we can just walk away from it.
Right. Yeah. Now I'm picturing like Harry Potter. I think it's like Neville's family. Like he doesn't show magic powers, so his uncle like hangs him out of the window and drops him to see if he has magical powers or something like that. Imagine someone doing that with Anakin from time to time, like just see how force sensitive he is.
There is a book from James Luceno, Darth Plagueis, that I haven't read yet, and I'm eager to have it and see if actually there is something about this experiment, even if it's a few sentences, and see what or how he did it, and why, as you said, he left the mother somewhere in a desertic planet.
Right.
Darth
Sorry, go ahead.
Plagueis by Jim Lucino. Yeah, no, it definitely dives into Darth Plagueis and Palpatine, being his apprentice and different experiments they're running, and then finding out other things that, Darth Plagueis is not killed off, early in Palpatine's career, but he's still around during the Phantom Menace era and with Darth Maul. So it makes you wonder about the rule of two.
Yeah.
Is that a legacy book or is that a newer one?
Yeah, it's a Legends book.
I was just curious how long ago that came out, because they're definitely, I was going to say they're experimenting with this idea, but they're experimenting with this idea of experimenting a little bit more in The Mandalorian, which I know you said you hadn't seen, but you at least knew the basic premise of the scientific experiments there. So, and I hope to see more. I kind of wish The Mandalorian was more about that and less about other things that would be spoilers if I said them to you. Because to me, that's the most interesting. The doctor has a patch from Camino on, the genetic cloning planet and the tie into the clone troopers and those sorts of things. What if you made a whole army of clones that were force sensitive? That's an interesting question.
Yeah, it is. And I think that knowing more about the biology of midichlorians will definitely help to reach this goal of creating an army that is more sensitive to the force.
Well, let's just talk about creating an army in general. What is the science behind making clone troopers?
It's interesting because at the time that the movies were made, there were, we know the Dolly as a clone, a chip that has been cloned, and the issues that were with this kind of experiments, because actually the clone was aging faster because the chromosomes were not regenerated. But since there was a real revolution in cell biology, which is the reprogramming process. So that's Dr. Shinya Yamanaka in Japan, who had the Nobel Prize for that. So when I was a biologist in school, the dogma was that once the cells are from stem cells and they differentiate into different kind of cells in our body to get more specialized, they cannot get back to their original cells, the stem cells. But he tried to break this dogma and was successful by using different factors we call transcription factors. There are proteins that will regulate gene expression in a DNA in our cell nuclei. And he found a set of only 4 factors that if you force the expression in a cell that is already specialized, it will actually reprogram its gene expression program and get back to the original stem cells. And then that was huge. Because
that is huge to make stem cells back out of regular cells.
Yeah. It was it's something I don't know if we realize. There is the number of applications for that is just huge. And then the time, so it was 2007, I think, and it was made with a simple skin biopsy. And because it's difficult to get skin, it's not difficult, but it's for the people who get the biopsy, it's not an easy thing that can leave a scar or get infected. So researchers try to find other kind of cells that we can use to reprogram them into stem cells. Just to get back a little bit, one of the force of this discovery is that when we want to study genetic diseases, we use cells in culture and try to manipulate them, forcing gene expression and things like that. Then we had those embryonic stem cells. From those embryonic stem cells, we still need to manipulate them in order to introduce a mutation or things like that, or correct it, in order to study some genetic diseases. But then, because of this reprogramming process revolution, we can, from somebody that already have a genetic disease, take the cells, reprogram them into stem cells, and differentiate them into the cells that we want to study: muscles, liver cells, heart cells, cardiac cells. And those cells, we carry the genetic information from the original donor. And that's where it was huge. What I was saying, that it was made from skin biopsy, and now we can do it from pea sample.
Wow. So basically, if they got Jango Fett to pee in a cup, they
Yep.
could take his cells from that,
Yes.
make Jango Fett stem cells,
Yep.
and then use that to then start making their template for a clone army. But they could also genetically manipulate it. So if he had any sort of genetic disease, they could knock that out.
Yep, definitely. So the pee is sterile because it comes from the kidney, which is a huge filter, so there is no bacteria. So we can keep them in culture and in the laboratory. And then a few cells that comes from the kidney, and that we can put them in culture and make them grow. So we have a lot of them, and then we reprogram them, and then get induced pluripotent stem cells, so those reprogrammed cells. And then from that, we can imagine a lot of things to do. And as you said, there is a possibility to introduce mutation to study disease, correct mutations, but we can also start to manipulate gene in order to manipulate some phenotypes traits. And that's actually what can explain how the Bad Batch was made with manipulation from the stem cells to get different kind of clone of Jango Fett and having these different archetypes that have their own specificity.
For those of us who don't watch The Bad Batch, like myself, and I got up to that part of the Clone Wars, but I haven't finished the series yet. Just a quick, brief synopsis of what it is that makes them the Bad Batch. Are they experiments that got wrong, or how is that explained?
The Clone Force 99, or the Bad Batch, they were a bunch of clones that were intentionally modified to have greater abilities in different ways. That Hunter, the leader, he has excellent tracking abilities. Wrecker is just super strong. Tech has increased intelligence. Crosshair is designed to kind of be a sniper, so he has greater vision and greater stability, I guess. Then there's the question of what is Omega? Who is Omega? That is a clone that is a girl, but she's also a clone of Jango Fett. And so we see those, and they have adventures together, mostly as they're trying to figure out how to protect Omega, but also do the basic things of make money to survive and also avoid the Empire. So yeah, they get their name from the clone known as 99, who was a defective clone in that sense, and he became kind of a gnarled old twisted man that ended up being a hero during the Battle of Camino, and was a mentor to a lot of the clones because he'd been around a while. And
okay, yeah, that's interesting, because I wasn't sure if they were like experimented on a purposeful Bad Batch or a accidental or something. So that's interesting.
They may not have come out exactly the way that they've been programmed. One thing you mentioned earlier, Karim, was that when they cloned Dolly, they found that because of the degradation in DNA, that they accelerated aging happened, and that's kind of what the Kaminoans wanted with the clones, was that they would grow twice as fast so they could be combat ready in 10 years instead of like 20 years. But that also kind of implies that later on they get older faster. And we see that in Obi-Wan. You see a old clone trooper, out on the street, begging for money, that apparently they don't take care of their vets.
Interesting.
Yeah. So that's for Dolly. What they did is they took enucleated stem cells and then put the nuclei from specialized cells inside it. So the reprogramming happened inside oocytes, but it was not as efficient to rejuvenate those chromosome extremities, and that's why there was this accelerated aging of the future animal. But yeah, I think that's also with the knowledge that realizator or scenarist had at the time that led them to this kind of, at least it's coherent with technology that was used at the time.
Yeah, that's really interesting to me, because not being a geneticist, when I think about it, that seems like one of the least realistic parts of the clone troopers is this accelerated aging. But if it's possible here on Earth now, that's just interesting to me, that like, oh no, that one's actually possible. What is, so what
that is actually more likely.
Right.
But the thing is that the aging is really at the cellular level, but from a developmental point of view, I don't think that you will in 10 years have a grown-up adult. You still have a kid, but who will have cells from an older person.
I see.
there is a need to have like a device that will increase the growth of those individuals in order to get them in 10 years fully adult and operational.
Okay,
Right.
so with Dolly, it was just accelerating the aging of the cells, but we have to figure out something else that would accelerate the aging of the organism of
Exactly.
the human.
Well, and again, this is just, because it's a fantasy movie, but it's also like there are
Sure.
probably species in Star Wars that mature in 10 years. Like if you had just chosen a different bounty hunter, maybe Bosk was as a strong adult in 10 years. And like, why didn't they just use him instead of accelerating aging? Anyway,
and for the species of Yoda also, they are growing very slowly in the contrary.
Right. You're right. In Star Wars, we already know that different species age at different rates because we see, Baby Yoda is 50 years old. Yeah. And that's, sort of, I guess I saw it coming, and I think a lot of people did, since Yoda's 800 or something like that when he dies. So yeah, it's already built in there. So again, why not just pick a species? Whatever. The Kaminoans probably thought of that after a couple years in, but it was too late. They'd already committed. So.
Well, maybe Palpatine had his long plan and his short plan. He planned it to live forever and, have his own clones that he could transfer into or whatnot. So yeah, maybe he was like, okay, if I can figure out what makes a Yoda type being age so slowly or age so long, I could get the longevity of that without having the green ears.
Right. Yeah. Interesting.
One of the things when they started animal cloning with Dolly and several of the others was the issue of it taking a lot of tries to get a successful implantation and growth of an organism that, it might take like 700 tries or something. Yeah. And they managed to get that number lower, but hopefully the Kaminoans aren't taking tons and tons of tries in their artificial wombs to get a baby Jango clone to start. Otherwise, they'd need a lot more tanks.
Yeah, I don't know if in books or in the TV show there was something about actually the growth, those embryos, and then kids of Jango's clones.
Yeah, I don't think anybody's touched on that, but maybe we should pitch that to them. That what happens to? I guess that would be kind of miniature cellular bad batches because they don't implant and they probably just get flushed down the drain.
Mm-hmm. Yeah. Yeah, no, I mean, it would make sense. And I mean, at least I get the impression, I don't know if there's more on the history of the Kaminoans, but I get the impression this isn't their first go round, right? They do this every 10 years, right? But
so they figured this out.
Right. So centuries or, millennia ago, they figured out, but it would be different, right? It'd be different for humans if that's what Jango Fett is than for the other species and stuff. So there would be some sort of like first viable product wouldn't be the first one you tried, I would assume. But yeah, they don't show that.
Yeah. But they do have the sense that they need the Jango template from the original Jango still, that they can't just keep some sample of his and photocopying that into clone troopers. They have to occasionally go back and get original samples. Is that something that makes sense, or is that just a limitation that they created?
Yeah, it's a limitation that they created because now you can, if you get the cells, then you can freeze them and put them back in culture, have a batch that you can renew pretty extensively. So once you have the cells, you don't need the donor.
Okay, because I was wondering that too. Considering how they fly around to various planets, it doesn't seem necessary to find Jango Fett there on Kamino, but it's interesting. I just can't imagine as Boba Fett being raised around, thousands of lookalikes and they're all going through this training or whatever. It must be a weird childhood, I feel like, for everybody there.
Well, I think it's something creative because we look at the cell lines that we have here on Earth, the cell lines from the cancer of Henrietta Lacks. Mm-hmm. That basically those cells are immortal because you can keep breeding them and they're still growing more and they're still the same.
Yeah, so they're not really the same actually. Because when you grow cells in culture, usually it's on a petri dish, which is made of plastic, and it's very hard, and that's something that is not natural for the cells. So it may induce mutations or chromosomic abnormalities that will just keep going inside the cells. And then if you genotype the cells between different labs, you will see differences because of how they specifically are making the cell culture in each lab. Once the cells from the original donor has been deployed throughout the world since decades ago, between every labs you will have huge differences from the original and between the labs.
That's really interesting. I never thought about how the different environment would into that. I was thinking about this the other day. I've been looking into, and I'm not going to do it. It's just, I'm obsessed with my dog and she's getting older. So I Googled, I know people rich people get their dogs cloned, and I was looking it up, and it's very expensive. And it sounds like it's also maybe just a very bad idea, because you're not going to get, this goes into the behavior, right, that we're talking about.
Definitely.
It would be my dog, but it wouldn't be.
Yeah, it will be your dog at the genetic level, but the genetic is not everything. It's largely influenced by the environment, by the experience of the living form. So the dog will not grow the same, have the same experience, so definitely will not be the same being.
Right. Yeah. And then it was and then can you clone that clone? Could I just have this dog for the rest of my life? And, it's $50,000 or something like that. So I'm certainly not going to do it.
We can clone you as well. And so you and your dog will be forever.
Yeah, I just, once I started Googling, gets into, is this even, an ethical thing to be doing in terms of, because there are still mother dogs, that these clones get planted in, and those sorts of things, and what kind of weird implications there are for that, and all that kind of stuff. So I'm certainly not going to do it, but it was just interesting to me that the science for that is there. Again, it goes back a little bit to the clones we were talking about. They say that they modify their genetics, I believe, to make them less violent, more likely to follow orders, those sorts of things.
More obedient, less independent thinking.
Right. So would you even need to do that genetically, or could you do that behaviorally?
Yeah, I think it would be linking the genetics to behavior. We don't have the tool for that. We don't have the knowledge. I don't know even if it's possible because of the influence of the environment, and because during the brain development, there is so much interactions with those thousands of billions of cells that will interact with each other and then pattern our behavior. I don't believe that by genetic modification we can very specifically move the cursor in order to have it the behavior of the person less violent, more following orders, things like that. That would be
so maybe Tanwi was just lying to say we can do this, but they're not really doing it. They're doing it through the nurturing part, having them learn the correct behaviors that they want without actually doing the genetic part. But they'll tell that to their customers to make it sound really cool.
Yeah, that's why you end up getting so many independent thinkers in the end, not just because it makes for an interesting story in the Clone Wars or something, but when you're talking about editing out things, I mean, there is CRISPR now, so
what is CRISPR?
Yeah, so that definitely, so something that could have been used for maybe for the Bad Batch, maybe accidents, but if they wanted to try things, the tools that will be more adapted for that is the second revolution in molecular biology now, with the works of Jennifer Doudna and Emmanuel Charpentier, is the discovery of the CRISPR. The CRISPR are like genetic scissors that can target a specific sequence in a DNA and induce a cut inside the DNA. The cells does not like the cuts, so it will repair it. Because we have our information is in two samples, so they will use as a matrix the other strand of DNA in order to repair it. But if you introduce another DNA under the form of a plasmid, it's like a circular DNA that you can introduce in the cell nuclei. Then if you have a large amount of them, you increase the chance to have the cells use those plasmids as a matrix in order to induce the repairing. When it starts repairing, you need just to have a few bases that are the same than the DNA strand that you want to repair. But then you can introduce new genetic information, and then you can either repair or induce a mutation, or you can also introduce almost like a gene sequence inside the DNA. anywhere in the DNA. And that was a huge revolution. It was really interesting because this comes from how bacteria that are used to make yogurt are fighting against virus. So actually, bacteria are prone to a virus attack because there is a lot of virus, and virus needs a host in order to replicate. And when a bacteria is infected by those viruses, they will actually cut the DNA of the virus in small pieces and integrate them into their own DNA in a place where they can make the equivalent of CRISPRs. So then if a new virus comes, these CRISPRs will activate and then just more efficiently destroy the virus. That's how the bacteria are protecting themselves, that has been used in order to create the CRISPRs. And before CRISPRs, there was also two other tools that has been developed, but they are very difficult to use, less efficient. And the CRISPR is today the most efficient way to edit the genome in a very precise manner.
So let me get this straight. So like that bacteria, the virus comes in and they basically cut and paste part of the virus DNA so that when another virus comes in, they can say, hey, I've got this virus DNA and I can use it to help fight this new virus.
Yeah.
And that's happening all the time in our yogurt.
Yep.
But then with the plasmid and CRISPR, which the bacteria is its own CRISPR machine, but when we do it, we are using the part of the CRISPR to cut, and then we can paste in. That's what the plasmids do. They're pasting in, but they got to make sure that they have maybe the same starting code, but then you can just drop in everything else, and that's kind of your paste thing. And your own cell just says, hey, this looks right. Plug it in, plug it in at the other end.
That's roughly that. Yeah.
Yeah, that's cool. Well, and so I know that we're not using it. Well, hopefully, I don't know. I actually don't know what humans are using this for. But when you were talking about the clones and being able to pull out aggression or independent thinking, those sorts of things, it's not that we don't have the tools to do that. It's that we don't know what lines of code say.
Exactly. This is a so powerful tool that the limitation is only our imagination.
Ooh, we could introduce frog DNA so that we can get the clones to lay eggs.
Life finds a way. That never goes well. You know this. That's really interesting. And I like that what you say about it's only our imagination, because it is one of those discoveries that I feel like is a turning point.
Yeah, definitely.
In sort of the history of the science, and everything later is sort of post CRISPR, like, oh, now that CRISPR exists, we can do this or that. Yeah.
I don't know. Every time I hear CRISPR, I think of that also being the name of the drawer in your refrigerator that you put like lettuce or something.
Yeah.
So yeah, I just have this image of lettuce every time someone says CRISPR, and
I assume it's a tortured acronym, but I don't actually know.
Yeah, it is. I don't remember it, but it is. It's something with repeat sequence and
Right.
Nobody actually knows what it stands for anymore.
Right.
But I heard there was a real life organism that's called midichlorian. Do you know anything about that?
It's actually interesting because a group of researchers discovered a bacteria that's actually inside mitochondria. And they found it in ticks, and it's a symbiote. So because the ticks are keeping them throughout their progeny, and it looks like it's important for them, and there is a beneficial aspect of having this bacteria inside the mitochondria, but we don't know yet what it is. And they called it Candidatus midichlorian mitochondriae.
Amazing.
Yeah.
So there's midichlorian bacteria inside the mitochondria inside the cells of this tick species.
Yes.
Amazing.
This is like more specifically located in the ovarian cells.
And it's symbiotic, so it's helping out the
Somehow,
mitochondria that are then helping out the tick.
Yeah. And the mitochondria must bring something to the bacteria, so the bacteria will not overgrow and destroy the cell. So there is a balance between them, and it's currently studied.
Wow, that's cool. I love this feedback loop of science and science fiction, George Lucas and the other people clearly knew enough or watched enough, Nova specials or something like that to know some of these things to influence their fantasy. But then also generations of scientists grew up on loving Star Wars. And so, of course, like if I got to name a species, first I'd name one after myself. If I got to name a second species, I would probably name it after Star Wars. So I love that. Well, were there any other topics we should talk about?
Maybe what I can say about more specifically with CRISPRs, just that when it was discovered, because of its huge kind of application that we can make, the scientists over the world agreed to not use it to manipulate embryos. Everybody is trying to respect that, but there was an issue a few years ago when Chinese scientists talked about his research and the fact that he gave birth to two twins, Lulu and Nana, that has been modified with CRISPRs in order to delete a small DNA sequence in the receptor that is involved in the entry of the AIDS virus inside the CD4 blood cells. And so it's something that we should know about and be conscious that manipulating living beings and having human beings born with this kind of mutation, modification, we don't know what will be the impact on the future of those person. It's nice to have thoughts about it in our imaginary world, but definitely we must be very cautious in applications in medical studies and trying to play crazy doctors.
Right. No, I remember hearing about that because it was only a few years after CRISPR came out, I think, right? So it was like humans just couldn't help themselves, dive right into the ethical. I mean, I wouldn't even call it a gray area.
Yeah, there is definitely a line being crossed there, and that's something we should be concerned about.
Yeah, and I think that person was like immediately sort of like ostracized from the science. Like everyone was pretty unanimous in this is not the right thing to do. But those kids, exist. So that's
followed and
right. Yeah, exactly. There'll definitely be more, I think, examples of this sort of thing in science fiction now too, because it always works its way. The science kind of catches up, and so the science fiction makes a bit of a leap. So
we're back to the more recent Jurassic World movies with the girl being cloned. And anyway, if someone was interested in going into molecular bio or cellular biology, how would you recommend doing that? What was your path to getting into this field?
So I followed the usual path at the university and going through a PhD. And I made my PhD in the liver field, studying metabolic liver diseases. But since I was a student, I was amazed by developmental biology. That's something that was just, for me, incredible that from one cells you can get organized into a very complex living form that is in harmony, and then having them with progeny that repeat the pattern and evolve with time. So I looked for a lab after my PhD working on this and trying to focus on the liver because it was my specialty. I found the laboratory of Professor Steve Duncan at the Medical College of Wisconsin, and I joined his team, and he was working on liver development in a mouse model. During the 5 years that I spent in his lab, we tried to establish a model in order to have a human environment to study the liver development.
And then tell us a little bit about transitioning out of academia and into the store that you run.
Yeah. So after 5 years in Steve's lab, I came back in France. I first started in a lab near Paris. I stayed 2 years there, tried to develop my project, but it wasn't a good environment for that. So I decided to look somewhere else and ended up in Nantes, a city located on the west part of France. And there I was able to continue my studies with studying liver diseases. What was difficult for me is the administrative environment that we have to manage as a researcher. I think after a while, we are not experimenting anymore. We are following projects, mentoring students or technicians, so which is nice, but it kind of take out the salt of the research. And it was a little bit too much. And my situation was not stable financially because I still needed grants to get my salary. And then I started to think about things that I would like to do. When I arrived in Nantes, actually, I discovered a convention called the Festival des Utopiales. So it's a science fiction convention, the biggest in Europe. And I had the opportunity to make some intervention with other artists. And it was a very nice environment to share ideas, to talk about science fiction, science, like we are doing now. And I really liked it. I had a chance to participate with this convention for several years. Now I'm inside the association, which is nice. And that's something that I wanted to do with the project I'm conducting now, which is the bookstore and game shop. Our store is specialized in imaginary literature, so science fiction, fantasy, fantastic, and combine this to games as well. And times to times, we are inviting authors in order to talk about their books and try to also find some science that we can discuss about. Because usually with a science fiction books, there is a vision of what kind of discovery could impact the society later. And I really like this, so I try to enhance this kind of features and discuss about it and make people around in our neighborhood to talk about it.
That's great. I love that.
Yeah,
idea.
that's really cool. Yeah, bringing both, science fiction to the community, but also the science so that people think about it, but it also then feeds back into the science fiction community because they're getting these ideas that you're bringing in scientists for.
That's awesome. And that's a lot of what I hear from friends in academia too, is that it's just such a stressful cycle.
It is.
So I really like the idea, and I'm inspired by the idea that you took a beat and decided, wait, what do I like? And pursued that.
So what's the name of your store?
So in English, it's called Portals. In French, it's Portails. So in bookstores in French, it's librairie. So it's library. It's more like somewhere where you can borrow books. But in France, librairie, it's where you buy books. And we call this Librairie Ludique in order to combine the books and the game. And Portals, for us, it was because a concept that is both in science fiction, fantasy, and fantastic literature. To combine this, and then also the fact that you enter in the store and then you just leave your things behind, enter like in the imaginary world, and then just find something that will help you to escape this world that is sometimes sad.
So if you're ever in France, go to Nantes, go to Portals bookstore, visit Karim, talk science fiction.
Yeah, I hope to make it one day. We'll post some pictures maybe in the show notes because you gave us a little visual tour and it looks like a magical portal. Definitely. I love that.
Thank you.
Well, just to wrap up here, what is your favorite Star Wars character?
That's a good one. I think maybe the favorite, because he's very mysterious, will be the droid R2-D2.
Melissa's favorite character, so you answered correctly.
This is your opinion, but there is also the correct answer, and that is R2-D2. But tell us more about why you like him.
Yeah, because I thought about it, especially with the midichlorian thing, because midichlorians are sentient microorganisms that better link between living forms and the cosmic force. But it looks like that R2-D2 also have this connection with the force. But it's not like a biological organism. It's more like it's a droid. So what is the link? And that's something that is interesting. And that's why I liked it. And also, it's an odd character and I like odd things.
It's a great answer. I like it. Okay, we'll put a link to the bookstore's website, but if you have social media accounts that you want to plug or anything else.
Sure, yeah. We have an account on the Instagram. It's Librairie Ludique Portails, also Facebook and Twitter.
Okay, yeah, we'll post those in the show notes for people who want to follow. So, well, I'm so glad we got connected, Karim. Thank you so much for coming on and telling us all this cool stuff about genetics and cloning and cellular biology.
Thank you. It was very nice, and I really like this kind of chat. Great. Thank you for inviting me.
That wraps up this episode of Star Warsologies. We want to thank our guest, Karim Si-Tayeb, and we want to thank all of you for listening. We also want to thank astrophysicist Dr. Roland Lehoc for introducing us to Dr. Si-Tayeb.
If you have an idea for a topic for Star Warsologies or know an expert we should interview, let us know at Star Warsologies on Twitter and Instagram or at starwarsologies@gmail.com. That's StarWarsologies. We also have that cool Facebook fans group if you want to talk with us and your fellow listeners. We definitely encourage discussion. It is called the Star Warsologies Podcast Fan Group. You can search for it on Facebook.
And please, please, please do us a favor and rate, review, and subscribe to this podcast on your favorite service.
No topic is off limits, even whether Arabesh is really an alphabet or just a font.
We are part of the Skywalking Network, where you can also find a variety of other great shows about Star Wars, Disney, and Marvel, including classic Marvel Star Wars comics, the Neverland Clubhouse, Resilience Squadron, and the flagship show Skywalking Through Neverland. They also have a YouTube show called Today in Star Wars History. You can find all of that at skywalkingnetwork.com.
Join us next time on Star Warsologies for our panel from LA Comic-Con called The Living Force, Evolution in the Star Wars Galaxy. James will be hosting, and I am a panelist along with some of the great cosplay for science scientists. So if you're going to be at LA Comic-Con on December 3rd, our panel is at 11 a.m. in room 303AB. Or check in about a week later, and we'll have the panel audio up as an episode. See you next time.